I'll give a
Okay.
friendly wave.
What was that, Brendan?
I said I'll give a friendly wave. Start.
Well, you can wave now if you like, because people are now joining us. Hey, everyone, we're just going to get started in a minute here. I'll just let folks join. Everyone's probably just going to join at the last second as they grab their lunch and stuff. If you want, in the meantime, you can say hey in the chat and let us know where you're joining us from. Maybe you're joining us from a specific lake or from a different city in Alberta. You can feel free to type that into the Okay, it looks like there's still people signing in. Recognize a couple names, but it's good to see a lot of folks that I don't recognize. Awesome. Okay, looks like it's slowing down a bit, so I think we'll just go ahead and jump into the webinar. So if you're wondering who the heck I am, my name's Brad. I'm the Executive Director at the Alberta Lake Management Society, and we're going to be co-hosting the webinar today with Alberta Health Services. Our organization is just a small charity based here in Edmonton, and we've been supporting the rec water or beach monitoring program for a few years now. We have a lot of other programs that work to improve our understanding and awareness of lakes and reservoirs and watersheds. So feel free to check out our website to learn a bit more about the other work that we do across the province. Today's webinar is just going to be an hour long, and we are recording this. The recording will be posted on our website probably today or tomorrow following the webinar. So you can always access it or share a link to folks who might have missed it if you want to remind yourself of what we talked about today, or there's someone who wasn't able to make it today. The participants' cameras and microphones are disabled right now, but you are able to chat with us using the chat feature at the bottom. Or if you have an official question, submit it through the Q&A button. You can see what that looks like on the bottom of my screen here. It's just easier for us to track your questions using that feature. If it's an immediate question, I'll throw it over to the presenter, but otherwise we'll probably just wait till the end of the webinar to get to everybody's questions. If we do have any technical difficulties today, just try and sign back into the same webinar using the link that you originally signed in with. And if we just can't solve the problem within like 10 or 15 minutes, then we'll just go ahead and reschedule and send everyone an email. So today we're joined by a few different presenters. Janine Legare is the Natural Recreational Water Coordinator with Alberta Health Services. She started her career as a health inspector working in both southern and central Alberta. And for over 10 years, she's worked in the North Zone as a frontline public health inspector manager and now provincial coordinator. So thanks, Janine, for joining us. And then we also have two members of the Alberta Lake Management Society team. We have Sarah Klimchuk and Brandon Coull. Sarah and Brandon are both recreational water technicians at the Alberta Lake Management Society. They're the ones who will be supporting the program by collecting beach samples, but also coordinating sample collection with local volunteers or site operators. And I have their contact information on the screen below. So I think with that, I will invite Janine to share her screen, and she's going to get us started with the official webinar.
Sure, sounds great. Thanks for the introduction, Brad. And we'll just get started. Make sure everything is displaying properly.
Yeah, I will let you know. It looks good right now. I don't think it's totally full screen yet.
Okay, we'll fix that. Thank you.
Yeah, there it
There
is.
we go.
Looks good.
Perfect. Okay. Thanks. So today we're going to be going over our beach monitoring program. So I'll use beach monitoring and recreational water monitoring interchangeably today. So we're focusing on cyanobacteria and Enterococcus, and we'll get into a bit of an overview on both of those parameters. So first of all, a bit of an overview of what we're going to talk about. We'll give you a bit of a background of how we got here. And then we'll talk about some health advisories, and then we'll get into the real meat of this presentation, which is our sampling procedure overview. And we'll round everything up with some resources that can help you out through your sampling of your beach water. So what you can see on your screen right now, this is the Alberta Safe Beach Protocol. And this is the whole basis of our beach water monitoring program. And it includes a couple of key things. So first of all, it includes our benchmarks for Enterococcus and Cyanobacteria, and those are based on human health criteria. And then the next important things that this document has, it has a couple of tools that can help operators identify and manage potential health risks at beaches. And it is available online. So let's focus on cyanobacteria right now. So cyanobacteria is also called blue-green algae, but we're going to call it cyanobacteria for the course of this presentation because it is a type of bacteria, and it uses energy from the sun for photosynthesis. And it's also really interesting because a lot of cyanobacteria can control their buoyancy. So this means they can move up and down in the water column, and that's so that they can access the best levels of sunlight for their growth. Now, they are naturally occurring in a lot of our water bodies, but there's lots and lots of different cyanobacteria. So as you can see on your screen here, they can look very different, but when we see them, we're seeing these blooms, which are like a big growth of cyanobacteria cells. And so these blooms, they can look like a lot of different things. So on the left side here of your screen, you can see it almost looks like grass clippings in the water, or on the right side, it almost looks like somebody dumped paint into the water. And here in the middle, it's sort of looking like globs on the surface. Now, usually we do see these blooms more often in warm water, but as you can see in that middle picture there, they definitely can occur in winter or cold water. And their colors can be varied. So blue or green or brown or pinky purple. And when they start to decay, they often give off a pretty strong odor, sort of like earthy or musty. Now, our concern with cyanobacteria blooms is that some of these bloom-forming cyanobacteria, they produce toxins, and these toxins can be harmful to the health of people, pets, and wildlife. And there are a few different health effects that you can have either by coming into contact with these blooms or by consuming or drinking water that contains these toxins. And so that can be something sort of more basic like skin or eye irritation, up to nausea or vomiting or diarrhea, or even as extensive as like liver damage. Now, unfortunately, these blooms can be quite harmful to animals. We do see on occasion dog deaths or cattle deaths associated with drinking water or eating bits of cyanobacteria. Now, these toxins from these blooms usually do break down within days, but sometimes they can stay in the water at lower levels for weeks after a bloom disappears. And the amount of time it takes for it to disappear really depends on local conditions, like how much sunlight and other conditions that might be found in a local water body. The type of cyanobacteria toxin that we are most concerned with in Alberta is called microcystin. This is a common one here. And now we know that our types of cyanobacteria that we see in a bloom can vary throughout the season, but we also know that the amount of toxin that's present in a water body can vary throughout the season. So because of that, we treat all blooms as though they are potentially harmful. Now, when we look back at the Alberta Safe Beach Protocol, there are 3 different situations that it states that water quality is considered unsatisfactory for cyanobacteria. So the first is if you see a cyanobacterial bloom, so if it's visually identified, then that's unsatisfactory. Or if you collect water samples and submit them for analysis, the first is if that microcystin toxin, if the concentration is over 10 micrograms per liter, that's unsatisfactory. And similarly, if a sample is collected and the cell count is over 50,000 cells per mil, then that's also considered unsatisfactory. So we'll switch gears a little bit, an introduction here to Enterococcus. So Enterococcus is a fecal indicator bacteria. And an indicator bacteria means that the Enterococcus themselves don't directly cause illness, but because this Enterococcus bacteria is present in like the guts or feces of human and animals, then if we have high levels of Enterococcus, that means we're likely to also have high levels of more harmful disease-causing microorganisms in the water. Now, these Enterococcus bacteria can make their way into the water through a number of sources. A lot of times we see it's from things like runoff or storm drain outlets, or treated or untreated sewage effluent, or through sort of natural sources like soil, sands, and plant debris. Now, the Enterococcus water quality benchmarks from the Safe Beach Protocol are a little bit more complex, but bear with me, and we'll get through the slide here. So the first thing to know is that where the Enterococcus comes from matters. If the main source is from either human or cattle feces, then people are more likely to get sick. So that means when we have mid-range Enterococcus, so between this 1280 to 6400 CCEs or calibrator cell equivalents per 100 mils, those samples are sent for microbial source tracking. And that's to determine if the fecal contamination is from human or ruminant sources. Now, so if we look at this overall, satisfactory samples are either ones that are in the low range, so below 1280, or in that mid-range, but do not have any human or cattle or ruminant fecal markers present. Whereas unsatisfactory samples are either those that are in that high range, so exceeding 6400, or in that mid-range, but do have human or cattle fecal markers present. And if you're having an unsatisfactory result, there's increased chances of illness like diarrhea, nausea, vomiting, or skin or eye or ear infections. Now, because we have limited resources, we want to ensure that we are putting those resources to the best use. So we have to select beaches for routine sampling. Now, what we use is the water quality history at a beach plus the bather use to rank beaches. So this lets us determine which sites are priority sites that are eligible to submit routine beach water samples through Alberta Health Services and through our lab partners. And so every spring, Alberta Health Services, we send letters to our priority site operators and we invite them to sample, since all the routine sampling is done by beach operators, volunteers, or by ALMS rec water techs. Now, our sampling season every year runs the week before May long weekend, and it ends the week before September long weekend. And ideally, we want our sites to be sampled weekly, but we know that there are local conditions or some logistics that might prevent that weekly sampling. So for instance, you have a kids camp that is only open maybe the start of July. Well, they're probably not going to sample in May and June. Makes sense, right? It's not accessible to the public at that point. So just a quick overview of our priority site process. So first of all, we have Alberta Health the ministry coordinates and oversees beach monitoring, including the selection of priority beaches, and they coordinate all of our sampling through our lab partners. And then we have Alberta Health Services, Environmental Public Health, who I represent, and we work with priority beaches to help facilitate sampling. We also respond to complaints or concerns about water quality, and that is at any beach, whether it's a priority site or a non-priority site. And then we have our samplers, and it's last on the slide, but it's definitely not least because this program wouldn't exist without our samplers. And that's our beach operators, that's our ALMS network. Like I said, that includes our rec water techs, the volunteers, the watershed groups. All these people help contribute to this entire program. So we did talk about those benchmarks, what means satisfactory and what means unsatisfactory water quality. So if we have unsatisfactory water quality, Alberta Health Services can issue water quality advisories. And so we have a water quality advisory for Enterococcus, or for fecal contamination. And that's the sign here on the top of the slide that you would see. And that sign would be posted at a specific single beach where the health concern is present. And then when the subsequent sampling occurs and samples are demonstrating that conditions have improved, we've received satisfactory samples, we could lift that advisory and remove that signage. Now, on the other hand, we do have our Cyanobacteria bloom advisory, and that's the sign here on the bottom of your screen. And because we know that cyanobacterial blooms move around the lake depending on wind, water movement, and we know that the cyanobacteria can move up and down in the water column, when we issue an advisory for cyanobacteria blooms, that stays in effect for the whole season. However, our messaging for cyanobacterial advisories indicates that any areas of the lake with no visible bloom can still be used for recreational purposes. And as a note for anybody that's sampling, routine monitoring should still continue after an advisory is issued. Now, if you are involved in collecting water samples, we really want to put safety first. So first of all, if the weather is not conducive to safe sampling, don't sample. If you're seeing, lightning off in the distance, or if you have high winds and you're seeing a lot of wave action, it's not worth it to collect a sample on that day. It can be collected another week or another day. And then please ensure that you're looking at the safety equipment that's included in our sampling procedures, which includes things like life jackets, chest or hip waders, and gloves, especially if you are dipping your hand into the water where cyanobacterial bloom is present, because that can contain skin irritating compounds, and we just don't want that happening to any of our samplers. And then making sure that you have access to soap and water or hand sanitizer following a sample collection. So we do have written procedures posted on our website. If you go to ahs.ca/water testing, you'll get to a page that looks like this. You can see we have our recreational water monitoring for beaches section here, and we have handouts that describe the sampling procedure. And same thing for cyanobacteria. Plus we have links to videos that ALMS produced, which walk you through the entire procedure. And these videos are excellent resources. I'm so pleased that we have access to these now. And we also have water sample drop-off locations, and I'll show you that at the end of the presentation here. So now I'm going to pass it over to Sarah, and I will mute myself, but Sarah can take over. Thanks.
Perfect. Thank you, Janine. So just getting started with the Enterococcus sampling here. There are two types of Enterococcus sampling. So that would be grab sampling and composite sampling. I'm only going to be talking about grab sampling today just because it's the default method of sampling for the priority sites. and only a handful of sites are eligible to do the composite sampling. So if you are a site that is eligible for the composite sampling, please feel free to reach out to any of us presenters. We're going to have all of our contact information at the very end of the presentation, and we'd be happy to answer any questions or provide more information about composite. So then just going into grab sampling here on the next slide. As Janine mentioned, we have all of our water safety PPE that you're going to need. So that's your waders and your life jacket and your arm length or shoulder length gloves. Other than that, for the Enterococcus sampling, all you're going to need are these 3 Prov Lab microbiological bottles. So to prepare for your sampling trip, all you're going to need to do is label these bottles with 1, 2, and 3, or you could use another labeling system such as N for North, M for Mid-Beach, S for South, whatever makes sense for you and for your site, just as long as you're consistent week to week and you are using the same labeling scheme on the requisition form. And so we recommend that you do this before, so you have your bottles prepared and while they are still dry, and just by using a Sharpie. Next slide, please. So the next step of sampling once you're at the site is to pick your sampling locations. To ensure the best quality data, we just request that you're consistent with the locations each week or each month, whatever your sampling schedule is. This will help us determine if there's any variability along the beach, and it assists with the investigations into contamination sources, like Janine discussed, the microbial source tracking. So it's really important to have your bottles labeled consistently so we can track down that exact location. You can ensure this consistency by taking photos at your beach site and labeling them, or by labeling a satellite image or a map. You just want to make sure that these sample sites are evenly spaced out and they cover the entirety of the swimming or recreation area. And we also recommend going from left to right every time, just to help with consistency. And like I said before, it might help to use fixed landmarks like the image shows: playground, mid-beach, and gazebo. You can go to the next one here. So once you have your bottles ready to go and you've walked to the first site on the beach, you want to go to about hip depth, and you're going to wait for the sediment to sample completely. And with a gloved hand, you're going to remove the cap from the first bottle, just being careful to avoid touching the inside of the bottle or the cap. And then as the first picture shows here, you're going to rotate the bottle downwards at about a 45-degree angle and submerge to elbow depth. Again, why those gloves are important: if there's a cyanobacterial bloom, you're going to want to protect your skin. So once you're at elbow depth and the bottle is fully submerged there, you're going to rotate it upwards as it fills, as shown in the third photo. And next slide, please. Perfect. So once the bottle fills to the indicated line, you can pull it out of the water, and then if it's overfull at all, you can pour off the excess. And at that point, you can reseal the cap, just being careful not to over tighten it. And then you're just going to repeat this exact process at site 2 and 3. So sometimes the water is highly colored or turbid, and this inhibits the samples, which means that the lab is unable to process them. These photos here are from Prov Lab, and they provide an example of samples that were inhibited. So those are the ones on the left, and ones that were fine, not inhibited, the ones on the right. So because of the possibility of inhibition at the lab stage, we ask that the samplers try to minimize the amount of algae or sediment when you're filling the bottle, just as much as possible and reasonable given the conditions that you're in. So before filling out the requisition form, there is some important information that is provided from Alberta Health Services in the letter that goes out to beach operators. Every recreational beach in Alberta has these 3 unique identifiers: the water body name, beach name, and access number. It's really important that when you use this information to fill out the requisition form, that it is unchanged, specifically with no abbreviations. So again, this information is available from the AHS letter that's sent out at the beginning of the season, and it can also be obtained by reaching out to one of us or contacting your local health inspector. So on the Prov Lab requisition form, you're going to record the date and time that the samples were taken, and you will also fill out the water body name, beach name, and access number that were mentioned earlier. Then on the left side there, the only checkbox that you're going to fill out is underneath the non-drinking water, and you're going to check off beach. And then you'll fill out the collector name and phone number. And then the box on the right that says collection site, that's where you'll be putting grab sample number 1 or grab sample number 2 or grab sample number 1 by playground, whatever scheme works for you. You just want to make sure that it directly matches the bottle that it's referencing. Then you're going to stick on one of the bright pink stickers provided by AHS on the bottom left-hand corner of the form, just underneath the remarks and requests section. And if you have any relevant comments about the site, such as a bird or fish kill, or a significant amount of bird feces, anything that you feel relevant to that sampling trip, you can note in the same box as the pink sticker. And then no other information is needed, so it's fine if there's a lot of blank spaces. That's how it should look for this. And very important to keep in mind that you will have to fill out one requisition form for each bottle. So for one beach, you're going to have 3 bottles with 3 separate correlating rec forms. When the rec forms are all complete, there is a sticker in the top right-hand corner with the barcode there. You're going to take that sticker and stick it onto the corresponding bottle for that rec form. And then just on the next one, Janine, there's a photo. So you're going to also stick one of the bright pink stickers underneath the label. And then we'll just go to the next one for submitting the samples. Once the 3 requisition forms and their bottles are completed, you're going to place each bottle in a separate resealable bag with the requisition form in the outside pocket. And then you're going to put all 3 of these bags and bottles upright in a cooler with multiple ice packs to keep them cold, and immediately have this transported to either your nearest public health office or directly to Prov Lab Edmonton or Calgary. And like Janine said earlier, there's the link to follow for the bottle drop-off locations. And then just to summarize everything, it's a lot all at once, but it's pretty simple. You just have the 3 bottles to prepare. You're picking your 3 sites across the beach, sampling at hip depth and then elbow depth, filling out the requisition forms, putting your stickers on your bottles, and having them sent to Prov Lab. So that's all for Enterococcus. We're just going to switch gears here into Cyanobacteria composite sampling. And with that, I'll hand things off to Brandon.
Thanks, Sarah. So before we get started with the actual sampling process for cyanobacteria, there's a bit of equipment that you'll need. So some of it is supplied by Alberta Health Services and some needs to be supplied by the sampler themselves. So what is supplied by Alberta Health Services is the requisition form, the ACFT bottle, the resealable bag, 2 conical tubes, blue gauze solution, a safety data sheet, and a plastic wine thief. And then on the next slide, so what needs to be supplied by the sampler is a large pail, thermometer, cooler, ice pack, and aluminum foil. For the ice packs, we usually like to have at least 3 in the cooler, just to make sure it can stay that cold temperature for extended amount of times, just in case. So on the next slide, to sample water for cyanobacteria, we use a composite sampling method, which combines water from 5 different sites along the beach at 2 different depths. So on the next slide, so note that sample locations should be consistent week to week and include 5 locations that are knee depth and 5 locations that are mid-thigh depth. So that will be a total of 10 samples that we're collecting. So I have a visual to kind of help you along. So you can kind of walk in the snake pattern to do it all at once instead of backtracking to keep it simple. But if you start left to right, you would start at your knee depth 1 location, and then you move up to the mid thigh depth location, and then you would just keeping your way over, and then making sure you're getting 5 different locations with 2 sites each for a total of 10 samples. And on the next slide, so once you've determined your sample locations, the equipment needs to be cleaned before sampling can begin. So you're going to rinse the pail and wine thief thoroughly in the lake about 3 times. You want to make sure that you are cleaning the water in the same body that you're sampling from, just to keep consistency and not to contaminate any samples that we do collect. So once rinsed, you can make your way to the first site to begin sampling. To do so, dip the wine thief slowly into the water to allow it to fill up from the bottom and to get an accurate representation of the water column. You're going to pour the water from the wine thief into the pail out from the top as pictured, or you can drain it from the bottom, but that does take a bit longer. Make sure to repeat this sampling procedure at all 10 sites. So once all sites are sampled, you're going to head back to land. You're going to find a level surface where you can work on. You're going to swish the water around to mix the contents, and then you're going to use that to fill the ACFT bottle 3/4 full and the 2 conical tubes up to the 49 milliliter mark. Make sure to leave at least a milliliter of space for the Lugol solution. Then you're going to add a milliliter of Lugol solution to each conical tube, or approximately 1 dropper full, which should be provided by Alberta Health Services. So Lugol solution is a preservative that contains iodine and potassium iodine. It is toxic for algae, so it lets us get an accurate representation of the concentration of algae in the water sample at the time of sampling. Lugols can also cause irritation and it stains really easily, so make sure to wear gloves during this process. And a safety data sheet will be provided to you to just make sure that you know how to handle it safely. So we need to now make sure that the bottles are secured and properly labeled. So you need to make sure that the caps are finger tight. We don't want to over tighten, same as the Enterococcus bottles. We're going to apply 1 laboratory requisition sample number label to the side of each bottle and a second label to each lid. The label doesn't have to be on the side of the bottle or the lid. It could also be on the top. It depends what's easier. It can be tricky to get them to stick on some of the bottle lids sometimes. Label each bottle with the water body name, beach name, access number, date, and time. And then you're going to wrap the ACFT bottle with tin foil to prevent sunlight from entering the bottle. So to properly ship it, again, similar to Enterococcus, we are going to place the ACFT microsystem bottle and completed rec forms in one of the resealable sample bags, and then 2 conical tubes in a second resealable bag. You're going to place the labeled samples in a cooler with ice packs to keep them cold during transport. And then again, you're going to deliver the samples to your local public health office as soon as possible after collection. So here you can see the filled out requisition form, which has a bit more information to cover than the Enterococcus forms, but it's still fairly straightforward. So the red arrow at the top points to the ID label. Each rec form will come with ID label stickers. Ensure these labels are put on the rec form at the top, and also all the bottles for the corresponding rec form. The other information you need to fill out is the water body name, the beach name, the access number, the GPS coordinates, the collection date and time. The time is written in 12-hour format, so make sure to use AM and PM. You need the collector's name, phone number, email, and their affiliation. And then you're going to check off the water source. For the most part, it's probably going to be a lake or a reservoir, but always double check just to make sure you're not making a mistake. There's also the sampling type and treatment. That is always going to be composite and raw for the microcystin samples. And then there is some visible inspections of the water that we have to do, like turbidity, color, and evidence of cyanobacteria bloom, you will make those decisions at the body of water. And then there is also wind direction, 24-hour precipitation, and water forecast. That information you can find on the Weather Network and the ACIS Alberta site. You just have to make sure that the station is the one closest to where you're sampling from, and the water temperature will be taken with a thermometer. So for turbidity, it's basically how cloudy the water is and how it looks. So the leftmost beaker is an example of a clear sample with no turbidity. As you go left to right, you can see how the samples become more turbid. It is important to be consistent week to week with your classifications. So in summary, you're going to pick 5 sites across the beach and sample them at knee depth and mid-thigh depth. So again, 10 samples in total. You're going to pour off the water into the ACFT bottles and the 2 conical tubes. You're going to add Lugols to only the conical tubes. Then you'll fill out the requisition form and put sticker labels on each bottle, and then drop it off at your nearest environmental public health office. That's all for the microcystin sampling. If you have any questions, feel free to ask me or any of the speakers now or at a later time. We'll be happy to answer. So now I'm going to give it back to Janine, and she can speak on the deadlines for laboratory analysis.
Okay, thanks, Brandon. Yeah, so I mean, you put some work into collecting your samples, and we really want to make sure that these samples reach the laboratories in the appropriate times. So each environmental public health office has different deadlines for when samples can be received, and generally it's Monday to Wednesday, but there are some offices that have different deadlines. So make sure you check with the EPH office where you're submitting samples. I'll show you in just a second here how to check that for your local office. But it's on this page, ahs.ca/water testing. So here's a few resources. I'll actually start off with the water testing since we referred to it last. So this is that information, the Sample Your Water page for AHS. And if you just scroll down to recreational water monitoring for beaches, you can see all of our handouts for the different types of sample collection. And it goes through all of it, including having those sample requisition forms like Sarah and Brandon referred to. The other thing I wanted to draw your attention, we do have the links to the videos as well, the sample videos that ALMS has put together. The other thing I want to draw your attention to here is this water sample bottle pickup and drop off locations. So if you click on that link, it takes you to Inform Alberta. And this has all of our offices that accept water samples. So you could enter your postal code here, click this button, and it will sort by the offices that are nearest to you. Sort of a cool tool to use. And so for instance, if we're looking at Acadia Community Health Centre that's located in Calgary, if we look over here on the right side of the screen, we can see some of the details. So the drop-off days are Monday, Tuesday, Wednesday, Thursday, and Friday, except beach samples. Beach samples are only accepted Monday to Wednesday. So you have to check these details if you are dropping off at any given office here. Okay, so we'll jump back to the presentation. And if you're looking for advisory, or sorry, I skipped this top one, so I'll go there. This is an environmental public health page, and you can see it has a bunch of different topics with drop downs. But if we go to the beaches drop down, we've put some useful resources here, including a link to the Safe Beach Protocol if you're interested, some My Health Alberta information, including information on cyanobacteria or blue-green algae, some information on swimmer's itch, plus all of our sampling resources here. Now, if you wanted to check out some health advisories, see what we have going on for that, you would just go to the AHS homepage, so ahs.ca, and then click on news, and right away we can see active health advisories there. And it's all sorted according to zone. So for instance, in North Zone, you can see there was a lot of boil water, so that's drinking water advisories, but there was a blue-green algae bloom advisory issued for Muriel Lake. And as our season progresses, we will post all of our advisories if and when they are issued on this page here. I jumped ahead there. I want to showcase the ALMS website as well, so alms.ca. If we click on that, and if we go to programs, we can go down to recreational water monitoring, which is what we're focusing on today. And there's information here about ALMS involvement, a bit about Sarah and Brandon, the text, plus links to these videos that ALMS put together. And these, as I said before, are excellent, excellent resources. And I really strongly encourage you to have a peek at those if you have further questions or want to review the procedure for collecting samples. And that brings us to the end of our presentation. I'll just leave it on this screen here, which has our contact information. So if you go she.recwaters@ahs.ca, you'll get to me. And then we have Sarah and Brandon's emails, rec.water or rec.water2@alms.ca. And that's it for our presentation.
Okay, thanks everyone. So we do have a few minutes left here. If anyone wanted to ask any questions, again, you can throw those into the Q&A. We'll just give folks a couple minutes to ask anything they like. Nothing came in during the presentation, so now is your chance to ask anything you like. And do take a minute as well just to note down the contact information that Janine has up on the screen there. There are no questions coming in.
So perhaps, Brad, if you don't mind, I will just point out one thing. So if you go to ahs.ca/EPH, short for Environmental Public Health, that brings us to our Environmental Public Health homepage. If you are ever out and about, you see something that looks like a cyanobacterial bloom and you want to report it, you're not sure if an advisory exists for that water body, we have this make a complaint, request a service, or ask a question link here, and you can click on that, and we would certainly welcome reporting of any sort of bloom. You can put your information here and submit it, and then our public health inspectors will get that and investigate to see if there's a need for an advisory.
Janine, is that the same location that someone would put a comment about if they felt they got sick from recreational waters, or a pet or something became sick after recreating? Is that an appropriate location for that type of information?
Yeah, I mean, if somebody themselves has gotten sick or thinks they've gotten sick, I mean, obviously, if it's urgent, you need to seek medical attention. But we also have HealthLink, where you can speak to a registered nurse and who can guide you through some of that health advice. So 811 is the number for HealthLink. But for sure, if it's, you know, your pet was at a water body and then started experiencing some symptoms, we definitely encourage you to take them to a vet. But if it's, you know, everything's sort of resolved, you want to report it to us, yes, absolutely, that would be the same way to access us.
Perfect. Someone in the chat has asked if the webinar will be available as a recording. Yeah, absolutely. We'll try to get that posted today or tomorrow. and that will be on the ALMS website, alms.ca, under our recreational water monitoring program that Janine is showing there. So thanks for that question. And if you are an operator and you would like to sample your location, but you're worried about not having access to the appropriate gear, definitely let us know here at ALMS, and we will make sure you're equipped safely so that you can perform the sampling. There is a question in the Q&A asking if there's an easy way to see advisories that were posted in previous years.
Easy, maybe. Yes, you can see them, that's for sure. So I clicked through that really quick, but I'll start at our homepage. So ahs.ca, that's our homepage. Oh, there we go, ahs.ca. And we'll do the same thing. So we'll go to news and then active health advisories. And then you'll see a link here in the subtitle called Inactive Health Advisories Archive List, and you can look there. So for instance, again, if I'm looking in the North Zone, I would just go down here, North Zone Archive, and if I wanted to check a specific lake, honestly, the easiest thing that I find is to search on the page. So Control F brings out the search bar, and you can search for lake or whatever lake you're looking for. And it does go back, I think, to 2012, 2011. There we go. So we have a lot of different advisories. Yeah, you can see we had a number of cyano advisories issued back in 2011.
Perfect, thanks, Janine. And again, if you have any questions, now's the time to ask them. Otherwise, you can always reach out to us at the contact information that was at the end of Janine's presentation. Janine, do you want to just pull that up one more time?
Yeah,
perfect.
absolutely.
Great. Okay, it looks like there are no more questions. So I just want to say thanks to everyone who was able to join us today. I hope walking through the webinar this way is helpful for everyone to kind of understand how the process works and understand the reasoning behind it. Again, we will post the webinar on our website. So if you want to share it with someone or you need to reference it in the future, you will be able to do that. You'll also find those helpful training videos on our website as well. Otherwise, I think I will go ahead and end the webinar. So yeah, I wish everyone a safe and enjoyable summer season and happy sampling. Thanks, everyone.